Hi everyone, welcome back to my channel. So in today's video, I want to talk some more about clinical trials as they're applied to testing gene therapies. I posted part one last week, and today in part two, I'm going to go over a few different concepts. One is on outcome measures, also power calculations, and statistical significance. So, so this is for a blinded trial. As I mentioned last week, for a medical treatment to be approved, it doesn't always involve a blinded trial, meaning that there's a group of people who receive the real treatment, a group of people who receive a sham treatment, then you follow them both for a while. The people in the trial and the people evaluating them don't know who's who, and they just, in a blinded fashion, note how the people are doing, and then at some later time, you see, who was actually treated, who received the placebo treatment, and then see, okay, is there a statistically significant difference between the real treatment and the fake treatment, such that it's couldn't have been chance, beyond some certain probability. Okay, so what does that all mean? And this gets into the concept of a power calculation. So say you have a certain number of people in your trial, and you run your trial for a certain amount of time. Okay, so in the group that isn't receiving the real treatment, you would expect their disease to just keep on progressing as it has been. In the treated group, you would expect it to, at the least, progress more slowly, maybe stop progressing at all, and if you're really lucky, their symptoms would actually improve. So then the question is, in each of those, three scenarios, slowed progression, stopping progression, and improvement of symptoms, how many people would you have to have in the trial, and for how long would you have to observe them in order to prove with appropriate statistical significance that the treatment is actually effective? Okay, so that's where power calculations come in, and the power calculations are, based on they're usually done beforehand, before the treatment is actually tested in the clinical trial. So you have to make some assumption about how the treatment might do, and you might say, well, we think it will maybe stop progression, and if it does that, then how many people would we need to have in the trial to prove that it's able to stop progression? Now, obviously, if it actually improves symptoms, then you are seeing a much bigger effect, and the number of people you were having in the trial would be more than enough to be able to detect that also. The other thing is an outcome measure. Okay, an outcome measure means something, some test that you have the people in the trial undergo, at various points, while they're being treated, or in the case of gene therapy, since it's a one-time treatment at the beginning of the trial, you're following them at different points in time afterwards. Okay, and so what you would want to see usually you do this in a natural history study, before you even start testing the treatment or maybe even develop the treatment, what is the typical rate of progression of patients with this condition over, say, 6 months' time, a year's time, 2 years' time. And then knowing, what the expected progression in the disease without any treatment, and also significantly, not everyone is going to progress exactly the same. Sometimes people can appear not to be progressing at all or even improving. A lot of times that isn't because they're really improving, but maybe they were tired from travel the first time that they were in the trial. They were sick. They hadn't done the test before, so the second time they're testing it, they're able to perform a little bit better. So there's going to be, a certain range of, progression among different people. So what you want to do in a natural history study is to say, okay, if I'm going to use a certain outcome measure, what is the average rate of progression that people with this disease might have over a year, and then what is the range of progressions, the variance in the progressions among different groups of people. Because if you just have one person and you're testing a treatment, okay, well, maybe that person appeared not to have much progression because it's the effect of the treatment, or maybe for whatever reason they just didn't appear to have any progression. So basically, the higher the ratio of sort of the average progression versus the variant compared to the variance in progression, the better an outcome measure that's going to be, and the fewer people you're going to need in the trial. Okay, so to put some sort of scaling laws on this, the number of the average, the uncertainty in the rate of progression, with n people in your trial goes as 1 over the square root of n. Okay, so what that actually means is that if you're trying to detect with statistical significance an effect of a treatment which is half as big, that means that you're going to need 4 times as many people in your trial as you would if the effect were twice that big. But if you can run a slightly longer trial, assuming that the rate of progression is linear, that the amount of progression over 2 years will be twice as much as 1 year, will be twice as much as 6 months, then running the trial for twice as long, all else being equal, means you will only need a quarter as many people. Now, in practicality, for various reasons, people don't really want to do a final, what's called registration trial, which means the final trial that, if it's successful, would get the treatment approved. People don't want to do that much longer than a year. So kind of a year is, in a practical sense, kind of the magic number that people look for. So, how many people can you do, would you have to include in your trial over a year to be able to show statistical significance? And that depends on the actual condition that you're studying or trying to treat. The faster that the natural progression is without treatment, the fewer people you would need, because the progression is going to be greater. So it's going to, if treatment stops progression, that's going to be a larger effect. Also, figuring out what the best outcome measure is and what series of tests will be most sensitive to people with the disease and their progression, and at what stage of the disease. For an example, a natural history study of limb-girdle muscular dystrophy 2B that I was actually in, found that for a specific outcome measure, you'd need somewhere on the order of 40 to 60 people. And one thing that depends on is the size of the treated group versus the placebo group, the untreated group. So are you going to treat half of the people randomly with a real treatment, or are you going to have treat 2/3 of them or 3/4 of them? Mathematically, the statistics work better if you have the two groups the same size, which means that only half of the people are going to get treated. On the other hand, no one wants to be in the untreated group because they all hope that the treatment will benefit them. So a lot of times people, to sort of sweeten the pot for potential participants, they want to have a treated to control group ratio of 2 to 1, which typically makes the total amount you would need in the trial about 40% greater. So anyway, that's basically one reason that you want to do natural history studies and test a lot of different outcome measures, because you need to understand the disease, understand what the best measurements are, understand the expected rate of progression, to be able to properly put together the protocol for your trial so it will be adequately powered, because the last thing you want is a treatment that will actually work, but the trial wasn't designed right, so it wasn't able to show statistical significance, which wasn't actually the treatment's fault, it was the trial's fault. Okay, so there's a lot more, to clinical trials. Hopefully, I've thrown a lot of terminology at you, but some of the important concepts that the size of the placebo versus the treated group matters. So even if you're in the placebo group in a clinical trial, you're playing a very important role. The longer you're able to run a trial makes it much easier. You don't need as many patients to have an adequate test of statistical significance. And it's really important to to identify the best outcome measures that you're going to use in judging the treatment's effectiveness in the trial. So with that, I'll say goodbye, and I will talk to you some more later about some more about clinical trials. Best wishes.